HNMT. HNMT stands for histamine N-methyltransferase. HNMT inactivates histamine by N-methylation. This gene plays an important role in degrading histamine and in regulating the airway response to histamine. The catalytic activity for HNMT is S-adenosyl L-methionine plus histamine, which yields S-adenosyl L-homocysteine plus N-tau methylhistamine. Histamine N-methyltransferase's molecular function is histamine N-methyltransferase activity. The biological processes are brain development, methylation, respiratory gaseous exchange, response to cocaine, response to immobilization, stress, hyperosmotic response, positive regulation of protein targeting to mitochondrion, response to amine, response to glucocorticoid, and response to interleukin-1. Histamine is metabolized by two major pathways, N-tau methylation via histamine N-methyltransferase and oxidative deamination via diamine oxidase. HNMT encodes the first enzyme which is found in the cytosol and uses S-adenosyl L-methionine as the methyl donor. The neurotransmitter activity of histamine is controlled by N-tau methylation, as diamine oxidase is not found in the central nervous system. A common genetic polymorphism affects the activity levels of this gene product in several red blood cells. Multiple alternatively spliced transcript variants that encode different proteins have been found for this HNMT gene. Diseases related to HNMT are nasal polyps, gastric ulcer, ulcerative colitis, septic shock, and other intestinal diseases. Potent HNMT inhibitors are diphenhydramine, amodiaquine, the antifolate drug metropine, and tacrine. HNMT is decreased in the brain of people with Down syndrome and increased in individuals with Pick's disease. HNMT plays the dominant role in histamine biotransformation and bronchial epithelium. If you have any concerns about this gene, contact a practitioner familiar with epigenetics and nutrigenomics. We hope that you've enjoyed this S&P Bit.